Today we'll be examining the effects of diet manipulation on pesticide resistance, specifically in mealworms. This is a question we've been looking at and practicing for in Biology 151 lab for pretty much the entire semester. We're utilizing skills that we've been kind of fine-tuned and drilled into our heads in our studio to carry out this investigation. Our overall research question is, does diet composition, carbohydrates, proteins, lipids, or a set control group affect pesticide resistance when looking at the organism known as mealworms? Does the diet have influence or effect on acetylcholinesterase or an ACHE inhibition when exposed to the pesticide naloxone? This is our pretty much in-depth question that we're looking at. And the research hypotheses for these would be: the null hypothesis is diet has no effect on acetylcholinesterase inhibition. The alternative is diet has a significant effect on ACHE inhibition. I'm not going to try and say that again. This is important to know because this can affect crops, financial issues, like how it affects the farmer or even the consumer when it trickles down. And knowing how diet affects mealworms shows us efficacy in ways that mealworms could possibly be more susceptible to pesticides when looking at diet. How this works is it kind of blocks the acetylcholinesterase that shuts off acetylcholine, which causes nerves to fire and contract. So this will lead to the acetylcholine continuously firing and eventually causing nerve death, which will lead to the death of an organism as a whole. Our independent variable are the diet types, the four right there. Our dependent variable is how much percent-wise is the ACHE inhibited. Our class sample size was about 56, not even about, was 56 total mealworms, and the method was the mealworms were fed a diet consistently for 2 weeks, and the ACHE assay was performed when looking at naloxone exposure. We use an ANOVA for this test and the statistical analysis because we're comparing means across 4 independent groups, which means there's 4 different treatment groups, and we're looking at the means of all of them. Our results overall was a 30.54% inhibition for the mean of all the groups, but we can see a big group of that is coming from the lipid, which shows a little bit more susceptibility, which kind of could be an inference or an educated guess for the future when we look at the statistical analyses. A standard deviation of 22, the sample size of 56, which would be also notated as N. Our minimum is 2.23, and our maximum is 88.58. But we're just going off of ranges, which I took the liberty of doing max minus min instead of having an extra column. The visualization we made was a box plot, and my name is here. The title is ACHE inhibition by diet, and our scale is from 0 to 80, but 100 is about 80 to 90, 95. We utilized the functions in RStudio to help us, and these codes are shown right here. But we have pink for carbs, blue for the control, red for the lipid, and purple for the protein, and the diet type is labeled on the bottom, just overarching x-axis and the y-axis. We can see the inferential statistics here, kindly in this box, which has been laid out really nice. Good coloring. With the degrees of freedom of 3, an F value of 6.746, and a P value of 0.00627. This is important because it shows that a specific treatment group, when compared to another one, is statistically significant inside of this data set somewhere. We ran an ANOVA, and the code for our studios here, included on the bottom, but we also ran a 2-key because we knew one was statistically significant. So we took our ANOVA and then ran a 2-key on it, and we realized that the only one that's statistically significant from the control group is the lipid, with a p-value of 0.0006213, which gives us information about our hypotheses, which means we reject the null hypothesis because there is a statistically significant difference. And this shows that diet significantly affects the ACHE inhibition. A biological interpretation, which could be seen as the lipid diet results in the highest inhibition when compared to the rest of them, and it suggests lipid-fed mealworms are more susceptible and not resistant. The control group also happens to have the highest natural resistance, which could be a natural resistance, but we don't really know why they're so good at resisting it. Lipids, this shows that lipids could interfere with the detox pathways of how they get rid of certain pesticides, or can also change enzymes at a cellular level, well, at like a DNA level. Some questions I would like to have is, can we try different lipids, like where the fat is derived from, maybe like a plant versus animal fat. So we can look at like pork lipids or something. And what happens if we keep this study going on for a longer time? And we can measure the actual survival rates and not just the activity, which means looking at individuals instead of just how much of the acetylcholinesterase reacts to the pesticide. I also wonder how other pesticides would work with this instead of just the naloxone. Do pesticides derived from different chemicals change how it is expressed, or the acetylcholinesterase expires?